A very good morning to my dear children of class 6, and I am happy to welcome you all to today's botany class. So in today's class we will be learning about a topic called as germination. So what are we going to learn today? Germination. This topic is from the lesson called the flower. In our previous class we have studied about the detailed structure of seeds, right? So you know what are all the parts in a seed. Now at some point seeds have the ability to develop into a small plant. This is not new to you because in your day-to-day life you must have eaten some fruits and thrown in your garden, and that seeds will slowly develop into a small plant. This you must be seeing for many years, I guess. So about this process, how a plant develops from a seed, that is called as germination, and about that germination only the whole class is going to be, because today's class will be fully about the topic called as germination, which you see in your day-to-day life. So before learning about the definition for germination, let us recall some concepts what we have studied in a previous class, because without knowing those terms you may not understand what I teach you. So in a previous class we have studied about the structure of a seed, right? So in a seed you have a part called as embryo. Do you remember? In this corner you will have a part called as embryo. What is this? Embryo of a seed. So this embryo has a special property, that is, upon favorable conditions, whenever the seed has favorable conditions or good conditions suitable for its growth, this embryo region will have the ability to develop two things. One is the root system and the other one is the shoot system. Do you remember this? We have studied in a previous class that embryo has the ability to develop into a root and a shoot. Clear? However, this is not always happening, especially in unfavorable conditions. This will not happen. Only when there is good condition or favorable condition for the seed, this process can happen. So this is about the embryo you need to know before you learn the definition for germination. So what is the definition for germination? As per your book, germination is defined as a process. Germination is a process where dormant embryo seed, what is the word dormant mean? Dormant or dormancy means it is not dead, however it is inactive in nature. See, seeds all, even for your food, you cook many seeds, no, like green gram all you cook and eat, but when you store in a container for a longer duration, say for example even if you store for 6 months, it is not germinating. Why? Because those seeds will be in dormant stage, which means again if your mother takes some green gram and soak it inside water, the next morning itself it will sprout into small root like structure, right? So that is germination process. So why when it is stored in the container alone it is not germinating, and why when you soak inside water it is germinating? Have you ever thought why this is happening? This is because it is always in a dormant stage, which means it has life, but it can become active only when you give suitable conditions for its activation. Say for example, when you give water to the seed, it will immediately germinate, which means so whenever the condition is unfavorable, the seeds will be inactive. That inactive condition is termed by the word called as dormancy or dormant. So just now I drew the structure of embryo, right? The embryo in the seeds I have shown you already. So the word given here is dormant embryo, which means embryo has the ability to develop into a root system and shoot system. However, it is in a dormant condition inside the seed. So the process when a dormant embryo inside the seed has the ability to germinate, that is, has the ability to develop into a young plant. What is a young plant? The seedling is called the young plant. What is seedling? When a green gram is soaked inside water, the next morning you see a white root-like structure, no? That is the seedling. After 2-3 days, what will happen? You can see 2-3 small leaves also. So such small plant is called young plant or seedling. So the process of germination is defined as a process where a dormant embryo seed develops into a young plant or a seedling. So this definition you need to memorize. Always memorize the definitions. Clear? So see here, there is a seed and you can see the embryo here. Can you see a green portion here? This is the embryo portion which can develop into a root. See, a root develops, and also after some days it can develop into a plant. So this entire process is called the germination process. About this we will be learning in detail in the upcoming slides. Usually, germination is different in monocots and dicots. What are monocots and what are dicots? Monocots means monocotyledonous plants which have only single cotyledon. In your first lesson, you have studied about this, right? Differences between monocots and dicots. What have you studied? The examples all you have studied. What was the example you learned? It was maize, right? So, the maize plant only is shown here also. So, you are going to learn how germination is happening in monocots, and also in the next slide you will be learning how germination is happening in the dicots. Actually the process is somewhat same, it is not much different, it is almost the same except there is one difference. I will be telling in the next slide that what the difference is. Clear? So in a monocot, that is the maize, what is happening? Suppose if you soak a seed into a monocot seed into a soil, what will happen? The seed will absorb the water. So when you pour water to the soil, the seed will absorb the water. This is the first step. Whenever you put a seed, that is a monocot seed, into the soil, it will absorb the water surrounding it. If it is available, it will absorb the water. Once it absorbs the water, what will happen? The radicle grows. What is radicle? In any seed, you have two parts. One is called the radicle and the next one is called the plumule. You have studied about this in the structure of a seed. If there is a seed, like this, two portions will develop, right? The shoot one is called the plumule. Do you remember? The shoot system is called the plumule, and this one will develop into root, and this is called the radicle. So this we have studied already. Then the plumule develops into a shoot system. What is shoot system? Shoot system is anything above the ground level. If this is the ground level, this is called the shoot system. Let me rub this. So here, which is the shoot system? This is the shoot system which is above the ground level, and this is the root system. So as I told you already, whenever there is a seed, it develops into two parts. One is called the plumule. See here, this is the plumule, the green region is the plumule, and this light yellow color or white color, whatever it appears to you, this is the radicle. So let me label. So here, which is the plumule? So this is the plumule. And which is the radicle? This is the radicle. Clear? So the plumule develops into shoot system, that is the system which is above the ground is the shoot system, and the radicle develops into root system. This one develops into root system, that is the region below the ground. And what happened to the cotyledon? By the time the root system and shoot system develops properly, the cotyledon dries. See here, the cotyledon is missing. Why? Because when the plant is grown to some extent, the cotyledon dries off in the case of monocot plants. Clear? So what have we studied? Whenever you put a seed into the soil, it will absorb the water in the soil, and the radicle grows first. First, which one grows? The radicle, that is the root system will grow first and it will rupture the seed coat. What is this rupture the seed coat? Which means the seeds have a seed coat. You have studied, right? In any seed you have a coat like structure and that is called as seed coat, and this seed coat is broken down, it is cracked and it is ruptured, it is uncovered. First it surrounds the seed, right? It no longer surrounds because it is ruptured. When it is ruptured, as soon as the radicle develop, it will rupture, and then the plumule, that is the shoot system, will develop, and finally the cotyledon will dry and the plant will grow as usual. This is about the germination in monocotyledonous plants. Next we will see about the germination in dicots. As I told you earlier, it is almost the same, the process is actually the same, that is the development of root system, shoot system, everything, except there is one difference. Let us see what the difference is. So next is germination in dicots. See, this is the example for dicot plants. So what happens in dicot plants? As usual, when you put a seed inside the soil, what happens? The first step is the seed will absorb the water in them and it will swell. Okay, what is swelling? Swelling is the seed will become bigger in size. Whatever you soak at home, either it be a green gram or black gram or chickpea, whatever you soak in water, after 6 hours or in the next morning, if you see, it will be bigger in size, it becomes swollen because it absorbs water. So after 2 days, usually in a dicot plant, the time taken for germination is 2 days. Clear? So after 2 days, the radicle starts growing downwards. Which is radicle? Can you see here? So this is the radicle. So first step is after 2 days the radicle starts growing in the downward direction towards the ground. Then what happens? The plumule, that is the shoot system, starts developing. See, after it is well grown down only the shoot system. Whereas in the case of monocots, what did we study? Immediately the root system and shoot system emerges, right? Radicle and plumule. Whereas in this case, if you see, for longer duration the radicle develops and then only slowly the plumule will develop. And also you can see one more difference. Can you see the cotyledons remaining attached? See here you have cotyledons remaining attached to the plumule. In the case of monocots, cotyledon was missing, right? Immediately root system developed, shoot system developed and the plant began to grow. Cotyledon fell off. That's what we studied. Whereas in the case of dicots, if you see, for longer duration the cotyledon will be sticking on. See, after that only the leaves will slowly emerge. After you have a big root system only, the leaves will small leaves will emerge and then only the cotyledon will fall off finally. So this is the difference between the monocot and dicot germination. The cotyledon is above the soil. See, it grows above the soil along with the leaves or the shoot system. Whereas in monocot plants it was missing, right? Immediately inside the ground itself it fell off. In dicot plants alone it will grow along with the shoot. It is above the ground level. Where do you soak the seed or plant the seed? Below the soil only you put the seed for germination, right? Whereas it will come out along with the shoot system. That is the major difference between monocot seeds and dicot seeds. The cotyledon is above the soil and after the leaves emerge only the cotyledons will dry. After fully the leaf grows taking the nutrients from the cotyledon, then only it will dry off. So this is the major difference between germination in monocots and germination in dicots. Next we will see about the conditions required for germination. So whenever you soak the seed, immediately it will not germinate. There should be certain favorable conditions given to the seeds, only then the seeds will grow. What are those conditions? There are three conditions. The first one is water, the second one is oxygen and the third one is temperature. So when you want a seed to germinate, you need to provide sufficient amount of water and then proper oxygen should be supplied and also the temperature should be suitable for the germination. Only then the plant will germinate. the seed will germinate. So let us see about the conditions in detail. So I told you, you have to water the seed when you want it to germinate, because the seed has a seed coat. I told you there is a coat surrounding the seed which will always protect the seed, and it is hard in nature, which will not let the plant to come out. That's why when you store in a container, the seed will not germinate. But when you pour water into the seed, the seed will become soft, as well as the seed coat. I told you the seed coat is hard covering, right? This seed coat, hard seed coat also will become soft when it is submerged in water for a longer duration, and this hard seed coat becomes soft and it will crack and it will open. It will no longer, see here, you can see here the brown region is the seed coat which has cracked and is separated from the seeds. If this is the seed coat is separated from the seed. It is no longer sticking on to the seed. So the seed coat absorbs and it swells, and then it cracks open. And then after it is open, only the root system, that is the radicle, and the shoot system, that is the plumule, will emerge. And one more important process is it initiates the enzymes. What does this mean? The seeds will have many enzymes in them, but again the enzymes, just like the embryo I told, dormant embryo. Similarly, the enzymes are also dormant enzymes, that is, they will remain inside the seed, but it will be in the dormant condition. It will not work. There are many enzymes in the seeds. That's why doctors advise you to eat sprouts. When you soak the seeds inside the water overnight, and when you eat in the morning, the enzymes are all activated and you get many enzymes from the soaked sprouts. Clear? So this water is the one which will initiate the enzyme. It will activate the inactive enzymes which is present in the seeds. Okay, what will happen if the enzymes are initiated? If an enzyme is initiated, it will act on the reserve food. Which is the reserve food in a seed? The cotyledon is the reserve food. You have studied about cotyledons, right? So cotyledons is full of food which is required for the germination of the seeds. So once the enzymes are initiated, it will activate the cotyledons to provide food. Then it will provide food for the growing embryo, which means the seedling or the young plant. So what did I tell you? When you provide water to a seed, the enzymes are activated, and the activated enzymes will act on the food which is stored in the cotyledons, and that food is helpful for the young plant, that is the seedling, to germinate or to grow into a plant until the nutrients are supplied from outside. So for a seed, do you add fertilizers all? No, right? The seed doesn't take fertilizers or nutrients from the soil also. Until it becomes to some height, until it grows to some level, it will just use the food which is stored in the cotyledon. Clear? So this is about the requirement of water for germination. Apart from water, the plants also need oxygen as well as temperature for germination. Why oxygen? There is a process in every plant called as respiration. What is that? Respiration. Similarly, in seedlings also, that is in the seed also, the respiration process will happen. Younger plants also will undergo the process of respiration. What is this respiration? Respiration is the breakdown of food. Which is the food here? The cotyledon has food, no? That food is broken down in the presence of oxygen to give energy. So out of respiration, what you will get? The product obtained from respiration is energy. So when there is energy, what will happen? The seed will grow. That is, the small seedling will grow from the seed. Then the stored food is helpful in giving energy. How the stored food is helpful in giving energy? Because I told you cotyledon is stored in the seed in the form of food, right? So it is the food which is broken down in the process of respiration which gives energy. This food in the cotyledon only is broken down, and out of respiration process you get energy, as I told you. So oxygen plus food in the presence of respiration process will give the product as energy, and this energy will help the embryo to grow. What is embryo? Embryo is the one which will develop into a root or the shoot, no? So this energy only helps the embryo to grow. For growth we need energy, right? So this energy which is obtained by a process of respiration in the presence of oxygen will finally help the embryo to grow into a root or a shoot. So this is how oxygen is helpful. What did I tell you? There is a process of respiration in every seed. Because of respiration only you get energy, because the respiration process needs oxygen. In the presence of oxygen the respiration process will give energy, and with the help of energy the seeds will germinate into a small plant, that is the seedling or a new plant. And the final factor for germination is temperature. Usually plant can grow in temperature between 10 to 35 degrees centigrade. So below 10 degrees also it will find difficulty in growing, and above 35 also usually the plant dries because of higher temperature, no? So when the temperature is between 10 and 35 degree, the plant can grow very well. Every plant will have an optimum temperature. Optimum temperature means the best temperature at which it can grow. The optimum temperature of human body is 37 degrees centigrade, because it is the temperature inside our body. But however we can survive below, even in places where there is ice also we wear jackets all and we still survive, no? Similarly plants also can survive from 10 to 35, but still there will be a optimum or a best temperature for every plant that will vary, because one plant may grow well in a colder climate, whereas like apple trees all, no, you cannot find easily in our places all, because it needs colder temperature to grow. So every plant will have optimum temperature. In that temperature only it will grow the best. In other temperatures it will find difficulty in growing. So temperature also is a factor for germination process. When you provide proper temperature only, the seed will germinate into a seedling or a young plant. So this is all about the factors which affect the process of germination. So today you will be doing an activity. Okay, so what activity? Activity for germination. Germination is the topic what we studied, no? So you are going to germinate some seeds in your house. Okay, so what you are going to do is take some cotton. Which cotton? There are two types of cotton called absorbent cotton and non-absorbent cotton. What is this absorbent cotton? Absorbent cotton means cotton which can absorb water. When you pour water on it, it will absorb water. Some will be like non-stick, no? Some cotton will not stick. Do not get that, or if you have at home also, do not use this or use that one, non-absorbent. You need absorbent cotton which can absorb water. Okay, so take some glass bottle or glass tumbler. Okay, use glass or even plastic which is transparent. You can use cups, plastic cups, all transparent cups if you have, no, that is well and good, you can use even that. Okay, but don't use plastic which is not see-through, which is fully covered, no, don't use, because you cannot see the process of germination. Use any glass container or a plastic which is see-through, like which is transparent. You can use that container. Put some cotton. Okay, so if you take a tumbler, put some cotton inside. Okay, put some cotton, then you can put any seed. If you have green gram at home, that is also well and good. So you can put some seed on the cotton. Okay, then what you are going to do, again put cotton above the seed. Okay, so take some cotton, put seed in between the two cotton, above also you have to put cotton, and then what you are going to do, pour some water. Add water, very little amount of water you will add, okay, because do not fill the glass with water, just sprinkle until the cotton is wet. Are you clear? Keep sprinkling water until the cotton is wet. Keep as such in a place where there is light. Don't keep under hot sun. Inside your house itself you can keep, where maybe near the window, or you can keep where it gets somewhat light. Not exactly the sunlight, light is enough, like it should not be in a dark room, that is what I mean. Okay. So what have we studied? We have studied that for the germination of a seed it needs water. So we have poured water, and also I have asked you to keep near your window where it gets oxygen, that is air, and also some light also it should get. So keep this setup as such. So after 1 or 2 days you can see the root system coming out, okay, like a white thread it will come out. So you can note the day, say for example if it is day 2, you can note it as your observation as day 2 and write what is root system called, radicle, no. So you can write as radicle on day 2, and then suppose in day 3 you get the plumule. What is plumule? The shoot system. If you get the plumule on day 3, in your observation write day 3 and write as plumule, okay. And then after 1 or 2 days you will get 1 or 2 leaves like this from your plumule, that is the shoot system. Then you can write maybe day 5 leaves like this, you can write in your observation note. And after you get leaves, no, take a picture and send it to me. Okay, so this is going to be your activity. You can use either mustard or green gram or chickpea, like these any grams or pulses you can use. Okay, so take a picture and send me. This will be activity for germination. Then your homework. Okay. For next botany class, you will have an online test from pages 24 to 35. This is lesson 2. So, pages 24 to 35 will be your portions for test. You are going to read this thoroughly, understand all the concepts, learn the definitions and memorize the technical terms. Don't be reading like a story book, because you need to memorize certain facts. It is not memorizing the whole paragraph or question and answers, but certain terms. Say for example, if I ask what is a shoot system called as, you should say that a plumule is the one which gives shoot system, like that. The technical terms all you should memorize. Clear? So prepare well for your exams. That is all for today's class, and until I meet you in one more class, you all take care and bye bye.